I may call the next speaker is Professor Hari Sharma. He's Professor of Neurobiology and Director of the Research Center for Central Nervous Injury and Repair at the University of Uppsala in Sweden. Welcome, and please, the stage is yours.
Thank you very much for this opportunity. I appreciate the invitation to talk, and I'm happy to listen to my previous speakers who explained everything regarding my speech. So that's why I know that you are hungry. I am very quickly doing my work present to you, and I can tell you only one thing. Blood-brain barrier is the gateway of all neurological diseases. On this subject, we are working since 1982. And I will show you that this is true. The second thing, I am very happy to listen my previous speakers, that cerebrolysin is strengthening the blood-brain barrier, and that also verify our assumptions made long ago. Well, there is no pointer, but I will do my best. Okay. Okay. These are my team who worked equally on almost all projects. Some of them are here. I mentioned, this is important, that whatever I am telling is my personal view, and it has nothing to do with the organizations or the governments with whom I am working. Traumatic brain injury is one of the great problems for our military community, because we are working for US Air Force and Department of Defense on research projects. And to them, traumatic brain injury is quite common. Traumatic brain injury is not only the injury by itself, but it also promotes several kinds of neurodegenerative diseases. For example, Alzheimer. It is well known that it causes, and that's why our veterans can get Alzheimer and Parkinson within 10 to 15 years. That's the big problem. I just show you some of the examples in Alzheimer that the problem comes even 20 years earlier, that you have the problem and it develops gradually over the time, and that's why it is very difficult to provide any therapeutic approaches. That's why we are not successful yet to prevent these long-known diseases. This is an example showing how Alzheimer progresses in the brain. And as you can see that, the Alzheimer disease, there is degeneration of brain organs. That is another example how ventricles are enlarged during the disease processes. Long ago, we know that our military community is vulnerable to these neurologic diseases. And also post-traumatic stress disorder, that is equally important like traumatic brain injury, as we have this disease in several veterans returning from various wars across the world. And nowadays, it is quite clear that traumatic brain injury, post-traumatic stress disorders, and even neurodegenerative diseases, there are many things very common. This is an example of brains. For example, the top one, A, is control level, and the D is post-traumatic stress disorder. You can see that spread of amyloid values very clearly. This is another example of showing post-traumatic stress disorder number C, how brain is stained with various problems. We have several regions in the brain, and each region controls diverse things. This slide shows you that the level of differences between frontal, cingulate, parietal, occipital, and temporal lobes, how they vary between post-traumatic stress disorder, traumatic brain injury, and a combination of them. Long ago, there was a research published in Proceedings of PNS, National Academy of Sciences, that its plasma level is very important in imparting diseases. And you can see that they have demonstrated this. In 2016, first study published, according to them, that in Alzheimer's disease, there is a leakage of blood-brain barrier. But as early as 2012, we published this report that in Alzheimer's disease, there is leakage of serum albumin in the brain, and cerebrolysin prevents it. So I'm very happy coming here to know, as my previous speaker said, that cerebrolysin is strengthening the blood-brain barrier. That's very good. Of course, we measured amyloid beta peptide, and we find increased. We are working on a project with the US government since 2004 on nano delivery of compounds or effect of nanoparticles on brain function. So therefore, our job is to label the compounds with different kinds of nanoparticles. Some of them are good, some of them are bad. And to study, can they prevent disease or can they penetrate well into the brain, and how long they can do this. So that's why when we have labeled cerebrolysin with titanium dioxide nanowires, that is most innocuous. Everything, even water, can be toxic. So titanium could be, but it is innocuous than many other substances that we are using. We can see the reduction in amyloid beta protein. The next example is tau, and that is also reduced when we delivered nanowired cerebrolysin. And that's why we believe that nano formulation of drugs in different amounts or various agents can really modify the disease states. And in this context, I just want to show you some experimental observations, how traumatic brain injury can exacerbate Alzheimer's disease brain pathology. Well, these are details how we did. So therefore, we are studying various components and compounds in the brain as a combination of nanoformulation. Can they improve? Of course, this is preclinical studies. Maybe it is related to the clinical or not, I don't know, but we are working on this area. For example, nitric oxide is also dangerous, and there are many reports came that it is involved in Alzheimer's disease. This is our model of brain injury, concussive head injury. And concussive head injury alone can produce brain trauma, as seen by pathological observations. and they are different in right and left half. When the injury is done on right, the left is much more damaged because of counter-coup mechanisms, well known. That's why the brain injury can change the neuronal networks. That is well known by my previous speakers and in this meeting. Not long ago, we have shown that nano delivery is the future in Alzheimer's disease, and we compared several things using the same compound as conventional therapy and nanowired delivery. We always find, together with the literature, that nano delivery has better effect of the agents. We use this titanium nanowires. This is US patent, and my friend is Dr. Ryan Tian, who did it. This is Dr. Ryan Tian and Dr. Wang. They have the US patent of titanium nanowires, and that's why we are working on titanium dioxide nano delivery. When people talk about stem cells, there are reports that you can also allow them on nanowires. They live longer, they can perform better. So that is one of the examples that everything can be nanowired or nano-delivered, including the exosomes, as you have heard very nicely by Michael Schopp. These are some experiments, and in our observations, blood-brain barrier find topmost priority to measure since 1982, because I did a PhD thesis on blood-brain barrier in stress. Why some people get disease, why others not? The link is blood-brain barrier only, in my point of view. So you can see that whenever the blood-brain barrier is altered, either amyloid beta peptide infusion alone or together with traumatic brain injury, they are changing. We have also used mesenchymal stem cells, nanowired of course, and you can see that the nanowired mesenchymal stem cells are doing good, and then we combine nanowired cerebrolysin and nanowired mesenchymal stem cells. The results are improved. The other key molecule is neprilysin, that is increased in traumatic brain injury, and sometimes it is also reduced because this is a network, and also in Alzheimer's disease, they are reduced. So that's why this is an example showing neprilysin merged with amyloid beta peptide and glial cells. When we have added nanowired neprilysin together with nanowired cerebrolysin, we find better improved effect in Alzheimer's disease model. And this is an example of hippocampus, as you can see here. Cerebrolysin and traumatic brain injury with Alzheimer, when we have given in combination, hippocampus is better preserved. Now I'm showing a few examples that this is amyloid beta peptide, concussive head injury alone, and it is more enhanced concussive head injury with amyloid Alzheimer's disease. And for example, nanowired cerebrolysin alone also significantly prevented deposition of amyloid beta peptide. But when we combine with nanowired cerebrolysin, mesenchymal stem cells, and antibodies to neuronal nitric oxide synthase, the effect is dramatic. means combination therapy is working better. This is an example of cells. The combined therapy prevented the cell damage in the brain. NeuN is a nuclear protein that can assess the function of neurons, and we can see that, nanowired cerebrolysine with mesenchymal stem cells and antibodies to neural nitric oxide synthase really prevented the damage to nuclear protein. This is an electron microscopy image, and you can see that the treatment is much more effective as compared to the untreated group. We have measured several things, and I can tell you one of the important things is synaptophysin. That is an important indicator of neural network, and that is enhanced. Of course, you can't see small data, but you can see here that the treatment combined with nanowired cerebrolysine and nitric oxide synthase antibodies together with mesenchymal stem cells has significantly improved the synaptophysin level in brain. So therefore, we believe in our hand, we are still working on various combinations of agents with antibodies and drugs who can be protective in neurodegenerative diseases or traumatic brain injury, and the investigation is still going on. We can believe that combination and nanoformulation of drugs and antibodies and different things are very important in future research, at least in preclinical stages. As I can tell you that not all nano-delivered compounds are effective. This is an example where you can see that in this side, there are many nano-delivered compounds. They are on this side. And on that side, most important compounds are there. Some are in between. So you have to find out which compound given nano-delivery is working and which is not. That is also very important. Why nanowire delivery works? We believe, based on literatures, that probably they enter into the brain, they penetrate with tiny holes in the endothelial cells, go inside the endothelial cells, and deliver their compound as like mini osmotic pump inside the brain. And nano delivery of nitric oxide synthase antibodies is recognized by US federal award, and we are happy. That's why we are using different kinds of antibodies, nano delivery in brain diseases to understand why and how they are functioning. So the point is, in our laboratory, we are working with various kinds of nano delivery of things in combination with different agents to find better neuroprotection, at least in preclinical stage. Who knows when it will combine with clinical conditions? That's the question. These are our organizations who support it, different organizations, and other friends that are not listed here. I am from Uppsala University in Sweden, and I thank you very much for your kind attention.